So let's begin. Share my screen. Can somebody confirm that you guys hear me?
Yeah, I can hear you.
Great.
I do have a question, actually.
Okay.
I'm looking at lab 3, number 2,
Uh-huh.
and
Question number 2?
yes. And I didn't have any feedback on why my score was the way it was, but I was hoping you could go over that question with us.
All right. You said lab 2, question 2?
Yes.
Describe the clarity of the nutrient broth after inoculating with sterile paper disc overnight and an incubation of 35 degrees. Is it clear or turbid? Let's go take a look. All right, so this is tube 2.
Sorry, lab 3.
Lab 3. Let me make it a little brighter in here. I just noticed my shirt wasn't totally buttoned. And you probably saw that when I opened up the window. So lab 3, there is no question 2. It's practice your microscopy skills. Or do you mean this question 2?
Yes.
I think this lab is still open, so I'm not going to give the answer to that. Let me shut this down to confirm that. Where's the zoom? There it is. Stop the share. Give me a minute there. I've got to get to the Clark website and log into Canvas. If I can't give you the answer, I will help you with the question, but I'm not going to give you the answer. And I think this lab is still open because I believe I just graded that this week. You cannot see the screen, correct?
Correct. I just didn't get any feedback on what I needed to correct, so maybe that's more of a private conversation.
Well, I would have told you only if it's wrong or correct. So I'm not sure what you're asking for.
Which question I got wrong?
You don't know which question you got wrong? Yeah, I should have given you feedback there. Let me look this up. So lab 3, oops, that's not the lab. That's the worksheet to the lecture. Yeah, lab 3 was the last one I graded, so that's going to be open until this Saturday. July 31st, you have until that time to make corrections. All right, now let me see. Katie, are you the one talking?
Yes.
Okay, let me see what I did on your sheet here. Oh, too many windows open. There we go. You're not Caitlin, are you?
No, just Katie.
Katie, okay. There's a Caitlin in the class too. Okay. Oh, yeah, I'm sorry. You're going to see what's wrong now. I thought I went through that in Made sure I submitted that. 2 is wrong and it counts as 2 points. Not sure where you got your field of view calculations. You can get the field of view size in the table in the lab module. So number 2 is the one you got wrong, and
So my field of view was wrong.
yes.
Okay.
Just a minute. Let me go to the modules. And yes, it'll help. This is lab 3. If you can see the screen here now. There we go. Oops, that's the wrong one. Go back. I don't want the worksheet, I want the module. And you don't know what I'm saying because you can't see the screen. Somebody has a chat message there. I can't see the chat message when I'm sharing my screen. So I don't use the chat. So if you want to ask a question, all right, never mind. So you can sort of see Lab Module 3. I'm just having a hard time showing it in here. There we go. The field of view has been measured for you in this table. I don't remember which lens the question was, but the field of view will be one of these right here, in millimeters and in micrometers. Any question about that?
Thank you, sir. I had a question about that too, but you answered it.
All right. It's the oil immersion lens, so the field of view size. Will be 0.18 millimeters or 180 micrometers. Okay, any other questions before we get started? If not, let me shut that down. That's the grade book. And I'll go back to the home page. And let's talk about what we're doing today. So today's the 27th. We're going to finish chapter 4 and hopefully start chapter 5. Note that you do have a quiz 2 on labs 3 and 4. And it will also cover chapter 3 and the end of chapter 2. And while I'm remembering, let me look. You cannot see the screen, is that correct? Can somebody confirm that, please?
Correct.
All right. I need to go into the grade book. Well, yeah, I want to go into the grade book and make sure that the time is correct. And I don't want to show you the grades to everybody, so that's why I'm asking. Quiz 1A, I mentioned this, was a shorter time length than I expected most of you to finish the quiz. And this is an experiment that we are working with, although one of the instructors has gone totally that way. And that is that if you don't have enough time to finish the quiz, you can't look up the answers. Or if you look up the answers, you fall further behind and you'll answer less questions. And so that quiz was designed like 16 questions and you had only 13 minutes to do it. So I did not expect you to totally finish the quiz. However, I gave you 2 points, meaning 2 questions in extra credit. So everybody got 6 points in extra credit. A number of students got 100%, and one student got over 100%. I don't know if that was 110%, something like that. So actually the scores on quiz 1 were pretty good for well, I don't know, your average student. Not everyone did well, obviously. I'm trying to look on here the time, and I'm sure not seeing it. So let me go into the edit. So quiz 2, I'll tell you how many questions there are. It wouldn't be numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Between 15 and 17 questions. And I don't see how long it will take you. There is a time limit. I'll make sure you have at least 1 minute and either 2 or 3 extra minutes. This is bizarre. It doesn't have a time limit. Oh, this is because this is quiz A I got. That's the problem. Quiz 2A doesn't have a time limit. Let me go to quiz 2B. There are 20 minutes that you have on this. And how many questions are there? I'm going to go into edit again to determine how many questions there are. There are 16 questions. Okay, any questions about quiz 2B? Let's shut that down again. Let me share my screen. All right, so you should see the screen. In the lab today, we will be covering, actually, we're going to be discussing the unknown project. There is no lab today. Any questions about any of this? If not, let's begin chapter 4. That was slow. So we had finished talking about the prokaryotes, and last thing we discussed is how prokaryotes form endospores. Any question about any of that? If not, let's move on to eukaryotes. You're seeing a basic plant structure in the organelles in plants and a basic animal cell with the organelles in plants. Many of the organelles in plants and animals are the same, like the nucleus. Actually, the vacuoles are found in both animals and plants. They are more common in plants, but you can find them in animal cells. The central vacuole, which that's not labeled that way, but it is showing you the central vacuole, is the largest organelle in a plant cell. In an animal cell, the largest organelle is the nucleus. Any question about any of that? Actual electron microscopic image of a plant cell. Well, plant-like cell, algae, and then a animal cell. This table is showing you a little bit about the, blow that up that way, of the different organelles between prokaryotes and eukaryotes, and a few other differences between the cells. Like, for example, there's a difference in the cell size, and the range will vary depending on the author. Actually, I'm not aware of any prokaryotic cells that are as small as 0.02 micrometers. I think the smallest prokaryotic cell that is viable is 0.4 micrometers, and they might be counting a cell that most people think is a non-viable cell, and the cell divides. Sometimes it can be as small as 0.02 micrometers, but I don't believe there's any viable cell that's that small. And then they go with the range to 2 micrometers. Well, you've seen that a different range in other places, and certainly prokaryotic cells can get much larger than 2 micrometers. Eukaryotic cells are typically larger, about an order of 10 times larger, and they can be much larger. The largest cell you've ever seen is the yolk of a chicken egg, and I know everybody's seen that. I've actually had a few students who have seen the yolk of a turkey egg, and that would be larger. However, very few of you have seen that. I've never seen the yolk of a turkey egg. The largest cell on Earth is the yolk of an ostrich egg, or I should say the largest cell on Earth at the moment. There are no nuclei in prokaryotic cells. There are in eukaryotic cell. Membrane-bound organelles are absent in prokaryotic cells. They're present in eukaryotic cells. Flagella can be found in both prokaryotic cells and eukaryotic cells. The flagella are different, but we're really not going to talk much about the differences of the flagella. There is a glycocalyx, which is secreted in some prokaryotic cells, and we call that a capsule or a slime layer. And there are some eukaryotic cells that have a glycocalyx as well. These are cells that lack a cell wall, so these would be either some protists or some animal cells that secrete a glycocalyx. The cell wall is usually present in the bacteria, but it's not present in all bacteria. There is one family of bacteria, we mentioned it, the Mycoplasma, that lack a cell wall. The archaea are a little bit different, and then there are some archaea that don't have a cell wall. But most prokaryotes have a cell wall. And then the eukaryotes, it depends on the kingdom whether they have a cell wall or not. Animals do not have a cell wall. Plants do have a cell wall. And then the protists, some do and some don't. The cell wall, if it's present in a eukaryotic cell, it's much simpler chemically than the makeup of the cell wall in a prokaryotic cell. Any question about that? For example, in plants, cellulose is the main molecule in the cell wall, and there's very few other molecules in the cell wall. The plasma membrane is surprisingly similar, oops, didn't mean to switch there, between the bacteria and the eukaryotes, and there's probably a reason for that, and that is that the eukaryotes appear to have borrowed the plasma membrane of the bacteria. And we won't talk any further about that, but their makeup is both made up of primarily phospholipids. The archaea have a different cell membrane as well as a different cell wall. The cytoplasm, this table is a little bit out of date. The author is at least 10 years out of date, and the author is saying that there's no cytoskeleton in the prokaryotic cytoplasm. That isn't true. They have a simple cytoskeleton, and the eukaryotes have a complex cytoskeleton. And then they have cytoplasmic streaming. I'll show you a little video where the cytoplasm moves around in the eukaryotic cell. Ribosomes, we've already talked about this, are smaller in the prokaryotic cell at 70S, and they're larger in the eukaryotic cell with 80S. And this table is telling you that you can find 70S ribosomes in the mitochondria and the chloroplast of eukaryotic cells. And we'll talk about that at the end of this lesson. The chromosome in the prokaryotic cell is usually a single chromosome, circular, and in the bacteria it lacks histone proteins. In eukaryotes we usually have multiple chromosomes. They're linear chromosomes, and they're associated with histone protein. Cell division differs. Prokaryotes divide by binary fission. Eukaryotes divide by mitosis. Sexual recombination, there is essentially none, but they can exchange, well, mainly plasmids. We'll talk about plasmids if you don't remember. And then in eukaryotes, they can engage in sexual recombination by meiosis. Any question about any of that? Let me shrink that a little bit. Eukaryotes have both flagella and cilia. Flagella is a long whip-like structure that the cell can use for locomotion, and the cell could have more than one flagella. Cilia are short little hair-like structures, and they usually beat in unison, and they're also a structure for locomotion. So if there's few of them and they're long, it's a flagella. If there are many of them and they're short, it's cilia. Remember, there are no cilia in the prokaryotic cells. And they're both used for locomotion. The cilia can also be used for moving stuff along the outside of the cell and then moving it to the surface or the mouth of the eukaryotic cell. We talked about the cell wall in eukaryotes. It's a simple cell wall. I'm only going to talk about plants, and they have no peptidoglycan. The cell wall of plants are made up of cellulose, and that's true of the algae and some of the fungi. I'm not going to talk about other fungi or the yeast. Remember, cell walls are absent in animal cells. They do have a plasma membrane, and they may be covered with a glycocalyx. Okay? Any question about animal cells? Eukaryotes have a cell membrane which is similar to the bacteria. The main molecule is the phospholipid, and it forms a bilayer. You can find proteins in the cell membrane, the peripheral protein, the integral proteins, and the transmembrane proteins. And remember, it's the proteins that move most of the molecules across the cell membrane. There are only a few that can move by diffusion, such as oxygen and carbon dioxide. The majority of the other molecules move across the cell membrane by a protein, and that can be either facilitated diffusion or active transport. So you can find transport across the membrane. You can find it in simple diffusion, and oxygen and carbon dioxide do that. You can have facilitated diffusion, and I gave you a very good example, and that is fructose only moves into your cells by facilitated diffusion, and then it can also move into the cell by osmosis, and that is only water. Remember, water is not a solute. All of the others, when we're talking about the molecule moving, we're talking about solutes. If water moves into the cell, it's crossing the cell membrane, so it's moving into the cell by osmosis. And then things can move in by active transport, where the cell spends energy to move a molecule against its concentration gradient. It involves an active transport protein and the expenditure of ATP. In eukaryotes, there is no group translocation. However, you can also move material into eukaryotes by vesicular transport, where something can come up to the membrane of the cell, and the membrane will then invaginate, and then the invagination will be brought into the cell by a vesicle. And then that vesicle can bring in materials into the cell. And that's endocytosis. And that can happen in eukaryotes. It does not happen in prokaryotes. Any question about any of that? Two types of vesicular transport. There's endocytosis, the movement of material into the cell via vesicle. And there's two types of endocytosis. Phagocytosis, where the cell will put out pseudopods, which will extend around the particle, engulf it, and then the cell will bring that into the cell as a vesicle, and this could be called cellular eating, phagocytosis. There's also pinocytosis, where the membrane folds inward on itself, and then greatly folds inward on itself, and then the membrane reforms, and then we get the membrane reforming on the vesicle inside the cell, and there we get a vesicle bringing the material inside the cell, and that's called pinocytosis or cellular drinking. Any questions about the two types of endocytosis? You can also have vesicular transport by exocytosis, where a vesicle brings the contents up of the cell up to the cell membrane, and then the membrane of the vesicle fuses to the membrane of the cell membrane, and then the material is just released outside of the cell by the secretory vesicle. Any question about any of that? All right, so let's talk a little bit about the cytoplasm. In eukaryotes, cytoplasm is the aqueous substance inside the plasma membrane, but outside of the nucleus. The cytosol is the fluid portion of the cytoplasm, but everything is called the cytoplasm. You can have cytoplasmic streaming. Let's see if I can get this video to work. Where the cytoplasm moves around in the cell. We might get a commercial there. Oh, that's awfully quiet. Yeah. Come on. So these green little things are chloroplasts, and you can actually see them moving around in the cell by cytoplasmic streaming. You can't see the central vacuole here, but it would be most of the cell and the inside of the cell. But the chloroplasts are moving around the cell this way. All right, any questions about that? The point is that the cytoplasm can be moving in eukaryotic cells. Come on. All right, we mentioned a little bit about the cytoskeleton. It's the cell structural framework, and it consists of 3 types of protein filaments. They make the backbone of the cell. And what they are just proteins, I don't know, running in the cytoplasm, connecting various parts of the cell, or I should say anchoring various parts of the cell. And in an animal cell, the cytoskeleton tends to give the cell its shape. That isn't true of a cell that has a cell wall, where the cell wall tends to give the cell its shape. Anyways, there are 3 proteins that make up the cytoskeleton: microfilaments, which I think is shown here, and then intermediate filaments and microtubules. The microtubules here are shown in green. You can see that the cell is basically this round shape here. You can't see the cell, you can only see the cytoskeleton there. And then this cell is basically this right here. And the cytoskeleton runs essentially throughout the entire cell. The blue part is probably the nuclei. Yeah, it is right there. All right, any question about the cytoskeleton? The complex structural framework running through the background of a cell, at least a eukaryotic cell. Prokaryotic cells do have a cytoskeleton, but it's much, much simpler than this protein network. Eukaryotes do have ribosomes, the site of protein synthesis. Remember that most eukaryotic ribosomes are ADS in size, they're larger, and the eukaryotic ribosomes are either free, found in the cytoplasm, or they can be membrane bound, especially to the nuclear membrane and the endoplasmic reticulum. And if they are connected to the endoplasmic reticulum, we call that the rough ER. It's called rough because of the ribosomes bound to the ER. And you should know that chloroplasts and mitochondria have smaller ribosomes of 70S size. Eukaryote cells have organelles, and by organelles I mean a specialized membrane-bound organelle or structure that allows the cell to be compartmentalized, and each compartment is a different organelle which is running or responsible for a different function of the cell. For example, the largest organelle in an animal cell is the nucleus, and its function is to hold the DNA and protect it. The organelles have characteristic shapes and sizes, and from them you can recognize which organelle it is. They do perform a specific function. It could be a function in cellular growth, maintenance, or reproduction. This is showing you the organelles, but this is all of the organelles, both the membrane-bound organelle, like a vacuole, and well, the flagellum, which is a structure of the cell, which is a non-membrane bound organelle. I think we got the cell membrane and the cell wall, which are structures of the cell, a non-membrane bound organelle. A ribosome, a non-membrane bound organelle. And generally, I will not use the term non-membrane bound organelle. I'll call that just a cell structure. And then when I'm using the term organelle, I mean membrane-bound organelle. And generally, I'll always try and use the term membrane-bound organelle to be specific or clear on what I mean. Any question about any of that? Let's see. You can find many of the membrane-bound organelles in both the plant cell and the animal cell, like the nucleus, the Golgi complex. There are some which are specific to one cell or another. For example, the central vacuole, shown there, and it's only labeled vacuole, but it's the central vacuole, is only found in a plant cell. Chloroplasts are only found in photosynthetic plant cells. Notice how I worded that. Chloroplasts are not found in the root plants. Cells, the root cells of a plant. And there is one organelle which is found in an animal cell and is not found in the plant cell. I think it's a lysosome. All right, any questions about any of that? You don't need to know the different organelles that are found in the plant cells other than perhaps the chloroplast. You should know that one. And you don't need to know that lysosomes are only found in animal cells. I'm not going to test you on that. All right, let's talk briefly about some of the organelles. The nucleus contains the DNA and protects it. The endoplasmic reticulum, there are two types. The smooth ER, which is, well, we'll talk about that later, what it does. And the rough ER, which is ER with ribosomes attached to it, making it look rough. The Golgi complex, which modifies and sorts proteins. A lysosome, which is a storage vesicle for digestive enzymes. And then the cell uses the lysosome to digest what it is consumed. We'll talk more about that later. Vacuoles can be found in both plants and animals. The central vacuole is only found in plants. It is a vesicle, which looks exactly like a vesicle. The difference is a vesicle is a organelle that moves in the cell, so it's moving something from one location of the cell to another. A vacuole is one that stores material for the cell and provides support for the cell. Okay, so it's a cavity that stores things for the cells. And a vesicle, once it's moved to its location where it's going to stop, it's no longer moving, so the vesicle can turn into a vacuole. And they do look the same, and they essentially play the same purpose. It's just that a vacuole is more for storage and a vesicle is for movement. But like I said, once a vesicle stops moving, it's no longer a vesicle and then it becomes a vacuole. On the mitochondria is the site of ATP synthesis and cellular respiration. We can say it's the cellular powerhouse of the cell. And mitochondria is the plural term. Mitochondrion is the single term, meaning if you're talking about one, you use the term mitochondrion. Chloroplasts, only found in plants, are the site of photosynthesis. Perioxysomes, they have enzymes for encouraging oxidation, and they're used against certain organic substances to oxidize them, or they have like peroxisome and other hydrogen peroxide and other peroxides, which the peroxisome can then release on something it wants to kill. Okay, a good example, I think we talked about the helminths. During a helminth infection, the white blood cell will come up to the helminth and then secrete noxic substances on the helminth to try and kill the helminth cells, and it's the perioxysome which can be released to release the toxic substances. But there are other organelles which can release toxic substances as well. The centrosome is involved in cell division. Boy, I went way too deep. It contains protein fibers and the centriole. The centriole is a structure of the cell that is used to make the mitotic spindle. So in cell division, the centriole makes the mitotic spindle, which then helps the cell undergo mitosis. Any question about any of that? So let's talk about the different organelles in a little greater detail, starting with the Golgi complex. Now let's go over here. The Golgi complex looks here and here, and it sort of looks like a stack of pancakes with maybe globs of butter. What happens in the Golgi complex is that vesicles fuse to the Golgi complex, bringing proteins that are made in the endoplasmic reticulum, so the rough ER, which then packages the proteins in a vesicle, and then the vesicle fuses with the Golgi complex. The Golgi complex then modifies these proteins, sorts them, and then packages them in a vesicle which buds out of the Golgi complex on this side of the Golgi, and you can see the buds forming on this side. And then these vesicles take the proteins to other regions in the cell. So that's how the proteins are transported by the vesicles. And these vesicles can fuse with the cell membrane, in which case they become secretory vesicles, where the cell can then dump the contents of this vesicle outside the cell. And a good example of that would be a teenager with raging hormones, and you're wondering why the hormones are raging in their blood. It's because the Golgi complex sorted the hormones and then put them in a secretory vesicle, which then went to the cell membrane and then released the hormones into the blood. Any question about any of that? All right, lysosomes, vacuoles, and transport vesicles all look the same. And as I mentioned, a transport vesicle, when it gets to a location in the cell where it wants to store the material, it stops moving, and we don't call it a vesicle anymore, we call it a vacuole. Our eukaryotes have an endomembrane system, which is a system or a conveyor belt for moving various cellular components, cellular molecules around in the cell. And I don't think that link works anymore. But let's see. Maybe it does. I got a new YouTube movie. Looks like another commercial. Come on. YouTube is big on giving commercials.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the trans face of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Moo Math uploads a new math and science video every day. Please subscribe and share.
All right, any questions about the endomembrane system? So there are 3 organelles which make up the endomembrane system, and that is the ER, especially the rough ER, which make proteins and then put them in a vesicle, shown here. The vesicle, it actually doesn't fuse on this side, it fuses on this side of the Golgi complex. And then the Golgi complex takes those proteins, modifies them. We're talking about chemical modification of the protein. We can attach different groups to the protein. We can change the protein, like removing an amino acid from the protein. And then tag the proteins and then sort them into packages which will bud out of this side of the Golgi apparatus in a vesicle, which will then take the proteins to where they belong in the cell. And that's the endomembrane system. Any question about it? All right. So this picture is showing you a vacuole in the cell, which looks like a vesicle, and a lysosome. Does anyone see the lysosome? There it is there. And you'll notice that they are essentially looking the same. So even though they look the same, they are serving a different function. The lysosome is involved in digesting things. The vacuole is mostly for storing things. The central vacuole, in particular in a plant, stores a lot of component molecules. And you can think of the central vacuole in a plant sort of being a giant refrigerator in the cell of a plant cell. And the cell puts its nutrients and other materials that it wants to use in the future in the central vacuole. But it also stores its waste products, like when you go to the bathroom. Instead of flushing it down the toilet, you put it in a bag, and then you put it in your refrigerator. And why the plants do that, store their waste and their nutrients together, I have no idea, but that's just the way plants do it. And remember, plant cells don't pee, so they store a lot of molecules, like waste, as well as nutrients, in their central vacuole. Any question about any of that? All right, no questions, let's move on. The mitochondrion is a special organelles found in eukaryotic cells, which are the site of cellular respiration. During cellular respiration, most of a eukaryote's ATP gets made. So the mitochondrion is considered the cellular powerhouse of a eukaryotic cell. The mitochondrion has two membranes, an outer membrane, which is shown best above in the artist's rendition, and then an inner membrane which is folded in on itself to increase the surface area. And here you can see these, structures are the unfolding of the inner membrane. Any question about any of that? Shrink that a little. The inner membrane is folded into cristae, which I'm not going to ask you about that, so you don't need to memorize it. But inside the cristae, you can find small DNA molecules and 70S ribosomes. Any question about the mitochondria? All right, if not, let's move on to the chloroplasts. The chloroplasts are the site of photosynthesis in photosynthetic cells. You only find, well, I should say photosynthetic eukaryotic cells. You only find them in algae and in photosynthetic plant cells. You do not find them in the root cells or in a plant cell which is non-photosynthetic. There are about a dozen parasitic plants, so they're not photosynthetic. The chloroplast is the site of photosynthesis. It too has two membranes, an outer membrane, shown in the green here, and then an inner membrane which is highly folded into what we call the grana. The flattened membrane sacs we call a thylakoid, and they're arranged into stacks called the granum. And what they all are is just the folding of the inner mitochondrial membrane. Not mitochondrial, chloroplast membrane. Any questions about any of that? Inside the chloroplast, specifically inside the granum, you can find the small DNA molecules and 70S ribosomes. So in the chloroplast and in the mitochondria, we have both DNA and ribosomes. Any question about any of that? why are there DNA molecules and ribosomes inside the mitochondria and the chloroplast? The endosymbiotic theory attempts to explain that. This is a theory to explain how eukaryotic cells evolved mitochondria and chloroplasts. And it also explains how they originated, and that's how what we mean when we say evolved. Let me, I probably should do paint. Let me try paint. So, a long time ago, we had bacteria. And we had we got it there, proto-eukaryote, which I'm going to abbreviate proto, UK for proto-eukaryote. And so this was a big cell. Let me see if I can draw a big cell. And the bacteria was a little cell, which I don't know if how good I'm going to be able to draw a little cell. Yeah, that's not bad. And the proto-eukaryote engulfed the little cell. So it became this over here. And for whatever reason, the proto-eukaryote did not initially eat the bacteria that it engulfed. And because it engulfed it, it was inside a vesicle, so we actually had two membranes, the vesicle membrane and then the bacteria's membrane, which I'm trying to show you right here. And we should have an arrow here because that's what had happened, but I don't know if I can draw that, so. Oh, that's not too bad so far, but this is going to be the hard part. Not bad. Not very good, but not bad for me. I'm not a good artist. The proto-eukaryote, for whatever reason, did not eat the bacteria initially. It was just keeping it around in the cytoplasm for later use. And inside the eukaryote, we had, I'm going to write it down here. Well, we can write it maybe in here. We had glucose molecules. And some of those went to the bacteria just by, well, not by diffusion because you're going across the membrane, but by transport across the membranes. And the bacteria took one glucose. Let me see if I can write that here. And by aerobic respiration, made someplace in the order of 36 to 38 ATP. This is a long time ago, so we don't really know how many. But the proto-eukaryote could not make that much ATP. The proto-eukaryote, I'm going to just abbreviate that P, proto-eukaryote. I need to write a lot here. Could only take glucose and make 2 ATP from it, probably by glycolysis and fermentation. All right? And then for whatever reason, let me see if I can draw that. Maybe because life was young, mistakes got made, the eukaryote released some ATP into the cytoplasm of the proto-eukaryote. We're just going to write ATP here. And like I said, it's probably that the cell life was young and so mistakes got made, and the bacteria made the ATP, and then some of it was released, and the let's see if I can extend that. Well, not really, but let me see. I'll move it forward. And this greatly confused the proto-eukaryote because it's now getting a lot of ATP. It only lost one glucose and then got many ATP. And the proto-eukaryote came to the conclusion that the bacteria was making ATP, much more ATP than the proto-eukaryote could make from one molecule of glucose. So the two formed a symbiotic relationship. Where the the proto-eukaryote gave the bacteria glucose and a home, and then the bacteria started making ATP for the proto-eukaryote. So that was actually a mutualistic symbiosis. Mutualism. So both benefited. Now they've lived together for so long that we do not find the bacteria free living on its own. So this species of bacteria we only find now inside a eukaryote cell. Okay? And they lived for so long that now, let's make another arrow here, that we now call them a eukaryote. Eukaryote cell, and a mitochondria inside it. And that's the theory, that a proto-eukaryote engulfed the bacteria, which performed aerobic respiration. The bacteria then started making, well, they lived in symbiosis. The bacteria started making ATP for the eukaryotic cell, and the eukaryote gave the bacteria a home and also glucose. And then they lived in symbiosis, and then they evolved into now what we call the eukaryote with the mitochondria inside it. Any questions about any of that? Well, surprisingly, this happened actually twice in some eukaryotic cells. In animals, we only got this happening once when the bacteria came into the proto-eukaryote and then later became the mitochondria of the eukaryotic cells. But in plant cells, they got the mitochondria from a bacteria, and then they got another bacteria which was photosynthetic, and this became the chloroplast in the plant cell. All right, any questions about any of that? Well, what evidence is there for this theory? Well, first, we can find that some cells, like this protistin here, can take up a bacteria, which the two will then live in symbiosis, and the bacteria will engage in photosynthesis and then make glucose for the protist. And the protist will survive will give the bacteria nutrients, for example, as well as a home. So the two live in symbiosis, and it is a win-win situation. So we do see this happening where a protist can take up a bacterium. When we look at the, well, this is a weak one, but when we look at the bacteria that gets engulfed by the protist, it'll have a double membrane because it'll have the membrane of the bacteria, and then it'll have the membrane of the vesicle that brought in the bacteria. When we look at the mitochondria, it has two membranes, an outer membrane and an inner membrane. So we expected two membranes if it was taken in by the proto-eukaryote cell. And then some other evidence. When we look at the DNA, we find that the DNA of the bacteria in the chloroplast is circular DNA, and it lacks histone proteins. And when we look at the bacteria DNA, it's a circular DNA which lacks histones, proteins. And the eukaryotes don't have that type of DNA. All their DNA is linear, and it's associated with histone proteins. And obviously the histones are a reason too. When we look at the first amino acid in protein synthesis for a eukaryotic cell, AUG codes for methionine. For a prokaryotic cell, AUG codes for formylmethionine, a related but a different amino acid. When we look at the first protein made in a mitochondria or a chloroplast, it's also formylmethionine. When we look at the ribosomes, the eukaryote has an 80S ribosome, the prokaryote a 70S ribosome, and the mitochondria and the chloroplast have 70S ribosomes. When we look at the way that prokaryotic cells divide, they divide mostly by binary fission. The mitochondria and the chloroplast also grow by binary fission. And the eukaryotes don't do that. When their cells divide, they grow by mitosis. And then the strongest piece of evidence, when we look at the sequence of the DNA, the eukaryote DNA, meaning in our chromosome, follows in the domain eukaryotes. The bacteria, the DNA will be in the sequence of the domain bacteria. And then when we look at the sequence of the mitochondria and chloroplast, where do you think they fall? Come on, somebody guess.
Can you repeat the question?
All right. Where do the DNA sequence of the mitochondria, where does it fall? Which domain does it fall in?
Eukaryotic.
That's a good guess, but no. The DNA of the mitochondria, when we sequence it, it is much more similar to the domain bacteria, so that the mitochondrial DNA is in the domain bacteria. Okay? Any questions about any of that? So there's very strong evidence for the endosymbiotic theory that the mitochondria and the chloroplast at one time were free-living bacteria that were incorporated into a proto-eukaryotic cell, and then that cell went in symbiosis, keeping the bacteria inside of it. And nowadays we don't find a free-living bacteria that we call a mitochondria. We only find that bacteria in our cells as a mitochondria. And the same thing with the chloroplast. Any question about any of that? All right, if not, that's it for chapter 4. Any questions about chapter 4 before I close it? I think I'm going to stop here, and then we'll have the lab at 6:30, where I will resume discussing the lecture. Any questions about any of that? All right, I'll see you at 6:30.